second one is the rarely appear.
So recessive means if for the appearance, for it to appear, you must have two recessive alleles, what we call alleles, okay? Remember that. Finish?
Planet square.
Yeah. What happened to the rest of the classroom?
They're sleeping.
Sleeping?
Yes.
Okay. Alright, so let's move on. So this chapter covers 19.1 to 19.4 and then 19.6, okay? So we'll look at, what we just discussed, how your genotypes is the basis for your phenotype. So genotype is the genetic makeup, while the phenotype is the physical appearance. We don't necessarily always see it, because for example in blood type is an example, you don't see the phenotype, okay? So we have understand how genetic patterns follow certain genetic inheritance follows certain patterns. We have remember the recessive and the dominant, and then we have different type of genetic inheritance. Okay, we'll start off today with what we call complete dominance, where the dominant allele is there, it will mask the recessive one. Okay, so yeah, and then we'll look at 19.3 other type. This is the type of inheritance that was established by Gregor Mendel in the 1800s, late 1800s. So he was a Christian monk. He was working on peas, and he established this basis for genetic inheritance. His two laws. One, the law of segregation, where he says that in the gametes, the genes, he didn't know there were genes at that time. He called them factors, okay? Because we didn't have, genetic, the DNA was found only in this last century. So in his time, we didn't have those. So he called them factors. So he studied that in peas, and he established the two laws. The law of segregation, where the alleles or the factors separate in gametes. That's why in the Punnett square you put them as, if you have you put the sperm or the egg, okay? Then you'd see the crosses. And he said that they reunite again during fertilization, okay? We'll talk about that later on. The other law, it was the law of independent assortment. Remember we studied last week about how the chromosomes can line up independent of each other when you have two different, more than one characteristics when you're looking at it. So we have 23 pairs of homologous chromosomes, okay, lining up in meiosis 1, remember? So they can line up either way, mother and father. You remember that?
That's a phase 1.
You forgot that?
No,
Yeah.
I remember it.
Yeah, so this is independent assortment, okay? So the alleles, the alleles can assort themselves independently of one another, unless they are linked, they're on the same, close to each other. We'll talk about that later on, okay? Then we have other type of inheritance, which is not, Mendel didn't establish, these are modern ones. Incomplete dominance, for example, skin color is incomplete because it's controlled by many genes, so what we call polygenic traits. Instead of one gene, when we're looking at, when Mendel did established his experiments, he only looked at, a particular phenotype that is controlled by one allele, okay? So these sort of incomplete dominance, where you see a variety of phenotypes, You don't get short and tall people, okay? You don't get white and black, okay? Skin color, you get a variation in between the two, okay? So this is due to incomplete dominance, okay? And then you have codominance, which both alleles, okay, they are expressed at the same level, 50%, like the AB blood type. Have you heard? Do you know about the blood typing?
Yeah.
So you will do this experiment in the lab, in the genetics lab as well, with the blood typing. But the AB blood type is what we call codominance. Neither A or B are 100% expressed. They're equally expressed, okay? So we'll look at other factors influencing the phenotype, like environmental factors. Okay, you might have the genes for being very tall. If you're not eating the proper diet, you're not going to grow very big. So environment also plays a role in the genetic patterns. Then finally, we'll look at some alterations in the chromosome number. So a very classic one is the trisomy 21 of the Down syndrome. You've heard of this one? So change in the number of chromosomes. Instead of 2 chromosome 21s, you have 3, okay? And there's a few others we will discuss there as well when we get to it, all right? So objectives is for your benefit, come to it later on, okay? And when you finish the lecture and see if you know your stuff. Okay, so before we start, let's define some terms. Some of these I think we already defined before. So when we're talking about genetics, we're talking about the study, okay, of inheritance transmission from one generation to the next. So from your parents to you, what genes they have passed on to you. When you study those genes, this is the study of genetics, okay? So the transmission of genes from one to the next generation, and inheritance is what is the actual transmission of the trait from the parent to the offspring. So when you inherit a trait, for example, he's got attached earlobes. He inherited those from his parents. You have unattached, you inherited it from your parents. So it's different genes that were passed on to you, okay? And we already know what genes are. Remember, genes are the functional units of DNA.
And inherited just from his father, or it could be from his grandfather?
Well, it's from
his family.
It's passed on through generations, okay? You can study, from the same family. But mostly when you're studying this, you look at in genetics, we look at the parents, okay? But the genes can come from obviously they come from somewhere.
From your mother.
All right. If you do a study, you can identify where they're coming from,
You can.
Yeah, you can. Sometimes you can use a genetic sequencer.
Okay.
Okay, you can sequence the DNA nowadays and find out where it's coming from. Okay, so genes are the structural units. Remember that sometimes they can code for proteins, sometimes they can code for RNAs, like the ribosomal RNA, tRNA, and messenger other RNAs. Okay. This word locus, okay, or plural loci. This actually refers to the position of the gene in the chromosome. Okay.
Other location.
Yes. So if you have a look at here, there's a chromosome. So the position of that specific gene is known as the loci. And we're not going to, there's actually a sort of a where they give it numbering to these genes, okay? We're not going to study that. It's based on the arms of the chromosome. You see the centromere here?
Yes,
So
sure.
based on the centromere, we can assign the long and short arm, okay? So there's short arm and long arm. So this genetic loci is usually a numbering assigned based on that, okay? But we're not going to study that, all right?
So this is chromosome.
This is a chromosome, yeah.
So
So,
this is the chromosome. Okay?
yeah,
All it's made of DNA.
yes.
What's the difference between all the chromosome and this region, the genes region?
Well, this is just the location of the gene, particular gene.
Okay.
So this could be, the gene for attached and unattached earlobes. There could be another gene here with a different loci, different location, okay? So we give them these locations because then we can find out where they are. When you do genetic studies, you have to know where these genes are. If they're close to each other, sometimes what happens is that they're passed on together, okay? Yeah, so when we talked about crossing over,
Yeah.
in crossing over, usually those two genes might not separate, okay? Because they will, when a crossing over happens, they will be they will move together, okay? So if you want to know what genes are doing, you have to know the location. If they're on different ends of the chromosome, they might not move together, okay? So it's important to know where the locations are, okay, for genetic studies. So the location, we call it the locus, okay?
Do we have to know like what does he mean by beta of autism?
This is, remember there's two types of chromosomes, okay?
Autosomes and
We
sex
have the
chromosomes.
autosomes and the sex chromosomes, okay?
Yeah.
So these are the 22 pairs, remember?
Oh yeah. Okay.
Usually, I mean, in the female it is homologous, the sex chromosome, but in male it's not, okay? So you will not see the same sort of thing because it's X and Y.
Yeah.
Doctor, he write here each autosome carry the same gene as the others. But the right one is red and the left one
About the
is
same.
just color or it's mean pink?
Which one?
And here.
The location here.
No. Pair of autosome, each autosome carry the same gene at the locus.
So
But the right one is red and the left one is blue. Just color or it mean something?
well, this is alternative versions of okay, of the same
Like
gene.
from mother and father.
Yeah, alternative versions, okay? So for example, you have the version for unattached elobes. He has the version for attached elobes, the gene, the same gene. Okay? So the gene is the same gene but in different versions, okay? To give, you know. Yeah, we will talk about that later on.
So,
Okay?
Doctor, what's the
So let's move back to here. So the locus is the position on the chromosome where the gene is found, okay? Then when you talk about genomes, we're talking about the total DNA, okay? The sum of the organism, okay? So in our genome consists of 46 chromosomes, okay? So the total genome refers to whether it's coding or non-coding, okay? So some of the genes, some of the DNA will code for protein, some doesn't code, okay?
Okay, the whole code it's called genome.
So the total, okay? It says here,
It's called genome.
yeah. All of the organism's DNA is called a genome, okay? Genome, okay?
And the gene for building.
So the total, it will include the 20,000 genes, all the DNA that is not transcribed, all the DNA that is transcribed but not turned into protein. So yeah, it's the total DNA, okay, in an organism.
And gene just for building protein.
So a gene is a functional unit of the genome, the DNA, okay,
Oh, okay.
one of them. And we have 20,000, 22,000 genes in there, okay?
And this is the main function for making protein, or how the main function?
Which one?
Genes.
Mainly they make proteins, but they also make some RNAs that are not translated, okay?
Oh.
So not all
RNA
genes are
translated.
translated. Okay, so we remember the karyotype. So the karyotype shows the chromosome arrangement. It is very important in studies where you're looking at alterations in chromosome number, because you can pick up by looking at this karyotype changes in chromosomes. So, you know, deletion sometimes, or additions, or even chromosomes missing, okay, or an extra chromosome. So this sort of karyotype is very helpful in identifying those type of diseases. So the scientists or pathologists use this to identify diseases. If they found some symptoms of it, they will look at the karyotype and see, yes, they will confirm it with that, okay?
From here, like down,
Anytime, you know,
down or
like Down syndrome, like Turner syndrome, like Klinefelter syndrome.
Diseases?
So, huh?
It have to be genetic disease?
Yeah, if there is a, sometimes there is a characteristic features of it, they don't need to do this, okay? But they need to confirm sometimes some cancers, some leukemias, they have alterations in some chromosomes, okay?
And doctor,
Like
most of the genetic disease, they couldn't find a cure for it.
well,
Like
I don't know all the diseases, so I don't know if they have cures for it.
Most of it.
Yeah.
Does the gene change when you are already reborn? So I mean,
Of course, you can't change it.
I,
But there is efforts now to do what we call gene therapy. They can change alter the genes in a particular organ. For example, there was one successful trial. There was one enzyme missing from the liver. They used the virus to infect the liver and transfer the correct sequence into the DNA of the cell.
so can you manipulate the
Yeah, but not as a whole organism. You could
Yeah, there are some.
There's one Chinese scientist. Chinese scientists, they made a genetically engineered child which was resistant to HIV. Do you remember that in the news?
Yeah.
But you have to do that at the level of the original cell, the zygote, okay? Once, start growing.
okay, what about the new child? When
Yeah,
you marry
yeah.
when he have children, you will separate his new gene.
Yeah,
all
but,
over
yeah,
And they may take
Yeah.
this.
I mean, it
use?
can be inherent. The new.
not fully, okay? Because the mother will provide one gene.
So we need two. One.
So, well, depending on what type of phenotype it has, if the dominant one is the one that is beneficial, then it will appear.
It's 100%
Yeah,
should be appear,
yeah.
or it's okay. It's maybe.
If it's dominant, it will appear. If it's recessive, it will not appear if the mother doesn't have it.
Okay, doctor.
We
what
need to
is
continue because we haven't got time. Okay? We've lost a lot of time. Alright, so the karyotype is shows the chromosome numbers, okay? And arrangement and characteristics such as the position of centromeres. You see sometimes centromeres are in different positions. So it's a very good tool to understand genetic diseases in humans. So remember we talked about the allele before. So the alleles is actually the alternative version of the same gene. Okay. So they have slightly different DNA sequences, which gives them that different phenotype if you get that particular allele. Okay. So in this case, if you have unattached ear lobes like me, this is a dominant phenotype. So I must carry either at least one dominant allele, okay? So I get two alleles, all right?
Yes.
So one of the alleles must be dominant, I must carry it. If you want to get attached, both alleles must be recessive, okay? So we'll talk about that later on here. So most of the traits in humans are controlled by, or genes are controlled by two alleles, but there is sometimes exceptions to that. Like for example, the blood typing, it's by 3 alleles. So remember the ABO blood typing. We'll do that in the lab, okay? So not always it's 2 alleles, all right? And when we talked about this dominant, so when we're talking about dominant alleles, we always put them in capital letters. Compared to the recessive, we put them in small letters, all right?
Recessive are the rarely.
Yeah.
Okay.
So we'll talk about the dominant and recessive, okay? Is it written here? No. So a dominant allele always appears, okay? When we're talking about complete dominance, so Mendelian genetics, a dominant allele always appears, okay? Even if you have one recessive allele, the dominant will mask the recessive one, okay? The recessive allele will only appear if both alleles that you received are recessive, okay? So a person with, for example, capital I and small I will have the dominant phenotype. To get the recessive phenotype, they need both A small. We'll talk more about that later on when we get to that monohybrid cross, okay? So, and when you, as I said, if you get 2 small alleles, you get the recessive phenotype. We call this homozygous, okay? Because both of the alleles are the same. So you can have both alleles the same in dominant as well if both are capital, okay? So you can get homozygous dominant, okay? Homozygous dominant or homozygous recessive, okay? Homozygous means both alleles are the same, okay? When alleles are different, we call it heterozygous. So capital I small I or capital B small b. So you can be homozygous dominant or heterozygous dominant, but you can only be homozygous recessive. Okay? Everybody understand that?
No. Okay.
So to have the dominant phenotype, you can be both homozygous dominant or heterozygous dominant. But to have the recessive phenotype, you can only be
Just
homozygous recessive, okay? And so we talked about the genotype. The genotype is referring to the gene, the makeup, the genetic makeup, okay? So of an individual. For example, for attached earlobes, that my genetic makeup. For unattached earlobes, because this is dominant, I could have any one of these, okay? My genotype for this particular trait. The genotype could be capital A or capital A small a, because this is dominant, okay? So that's the genotype. I can have two genotypes for this phenotype, all right? You understand that?
Yes.
But, so for one phenotype, I have two genotypes. Now let's have a look at him. He's got, do you have attached earlobes? Yes, so is it attached? Yep. For attached earlobes, there is only one genotype, okay? So the genotype is small a, small a. So genotype is talking about the genetic makeup, okay? While the phenotype is the outward physical appearance. So the my phenotype, say, is homozygous dominant, okay, for attached earlobes. That's my phenotype, genotype. But the phenotype is unattached earlobes, okay? The phenotype, okay? So the outward appearance. Genotype, say, homozygous dominant. The phenotype, unattached earlobes. The outward physical appearance, all right? So the genotype is based on this, on the genes, the alleles, all right?
Okay.
While the phenotype is the physical appearance, so like hair color, eye color, skin color, blood type, all these things are the phenotype, all right? The genotype is the genetic makeup, okay? So we show it by the alleles, all right?
So the genotype makes the phenotype.
Okay, yeah.
Doctor,
The genotype is the basis for the phenotype. Without the genotype, you're not going to have the phenotype. So I'll tell you, my genotype, for my unattached earlobes is A, or it could be A, small a. But the phenotype
It must be
is unattached earlobes. Okay?
But doctor, always we consider the attached one is a small.
So in this particular trait, yes.
Because I want to know how we can-
Different traits are different, okay? For example, so in this case, that the unattached is dominant in your fingers. If you have short fingers, the short fingers is dominant over long fingers. How many people here have short fingers?
don't know. How?
Do you have short fingers? Short, like that. Yeah, this is dominant. Long fingers are recessive, okay? So if he's passed on just one of the alleles to his children, they will have short fingers. Because it's you see, dominant recessive doesn't mean that it's more common. Okay? You see, a recessive trait is more common than a dominant one. Here, a dominant, most people here have unattached earlobes. It's dominant, okay? While the attached is recessive. So it's the opposite. So recessive and dominant has nothing to do with prevalence in the population.
It depends.
So it depends on the environment.
It's only to do with the genetics, whether, yeah. So it's nothing to do with the prevalence in the environment. You see, his fingers are short and it's dominant. My finger is long, it's recessive.
How could we say this is recessive
Huh?
or this is dominant?
Huh?
How could we see this is recessive or this
Because of the experiments that Mendel did, okay? And now we have modern genetics, we understand it, okay?
Okay, doctor.
If you carry one of the alleles in the phenotype is shown, it is dominant.
Dominant.
If you carry one of the alleles,
Yeah.
one version of the gene, it's immediately, it's dominant.
Doctor, the environment affect the gene?
Yeah, we'll talk about that later on. Okay, so genotype is the basis for the phenotype. Genotype, the genetic makeup, okay? So like when we designate with capital or small i's, and the phenotype is the outward appearance. We say he's got black hair, he's got blue eyes, he's got attached earlobes, he's got unattached. This is the phenotype, okay? Now these are some of the common ones that we see in humans, and these are all complete dominance, okay? So here, you see here the widow's peak. Widow's peak is dominant over straight hairline, okay? So to get widow's peak, you need these two genotypes. To have a straight hairline, you have to have these recessive, okay? Here, as I said, the attached earlobes, okay? You need to have either homozygous dominant or heterozygous dominant to get unattached earlobes. If you have the attached, you need the recessive, okay? You will be homozygous recessive, all right? So this is the genotype, this is the phenotype, all right? We understand now the difference between genotype and phenotype?
Yeah, I know the different ones. Why you said if you want to attach, you need heterozygous, and if you don't want to attach, you need homozygous.
Because
How can you know that?
this is dominant.
This is my
Okay.
question.
Okay, this is dominant.
How did I say this is dominant? Why?
This has been established, okay? Through genetic studies, okay? Like, I'll tell you what Mendel did before. You will know what it is,
And
okay?
they classified if it's attached, it's
Yeah.
dominant, and
We already know these characteristics are dominant, okay?
it's
And
classified.
recessive, it's already classified.
Okay.
Okay. So it's classified.
These studies have already been done. We already know this, okay?
Yeah. Okay.
So I'm just telling you what
Yes,
the
yes. No,
So
yeah.
we already know if it.
we already know this,
Yeah,
okay?
So they're already classified.
If
Dominant
there is a
and
new
recessive.
trait to identify it, you have to do the testing on it, okay? Crossing over, like Mendel did the monohybrid cross. Yes.
So
The
you know
the
it's
pea,
peas.
the
The
peas
peas.
and the pea flower, yeah. All right, so
Okay.
as I said here, you see the short fingers, this is dominant over the long fingers. To have long fingers, so most people here are recessive for the fingers, okay? They have this. They're
Okay.
homozygous recessive.
Yeah.
To get the long fingers, you have to be heterozygous, homozygous dominant or heterozygous dominant, okay? So now you understand that it doesn't necessarily mean that if you have a dominant genotype, this is prevalent in the population, because both dominant and recessive can be common in the population. This final one is freckles and no freckles, little dots on your face, freckles. This is freckle. To have freckles is dominant over no freckles. Okay, so these are some common traits. So the phenotype is not always controlled by the genotype. Okay, you might have the genotype for being tall, but you're in an environment in, say, in Africa or somewhere where there's limited food, and you're not getting the proper nutrition. You're not going to be very tall. Okay. So environment can affect the traits as well. Okay. So let's go and see what this is all about. How Gregor established these genetic.
Doctor,
Yes.
why the
people.
Asian people they have this tiny eyes? It's gene or what?
Well, it's gonna be genetic. Yes.
And it's
Dominant, there
dominant
is no
or
way to-
recessive?
Well, I'm not sure about that one.
It's always
There's lots of features.
No, because
thousands,
they
but
take
we know
another
everyone
one,
is dominant
another
or
one,
recessive.
Chinese.
No, but we
So it
say
will
it's not-
prepare
it's not
system.
anything in population. If it's dominant. or
It's
recessive.
if you take
مرات
an
تلقى واحد
Asian one.
مرات تلقى واحد ثاني متزوج.
I don't know.
And this is because the environment, they take their eyes from the sun or
It's
what?
recessive.
Why they are like this?
always like this. There is no way to see someone.
And
Because
they
they take from each other, so it will be recessive.
Yeah. So Allah created them like that.
أيوة, it will be شو اسمه.
Remember the Quran it says Allah says, I
Homozygous.
created you
Homozygous.
into tribes not because one is better than the other one.
Because you can know each other.
You can know each other. And the best one among you is
The one who has
taqwa.
the.
We already know that from the Quran. Allah created everything in different communities, into different tribes.
Scientific.
But I don't know what. There's a genetic basis for that. So we have to. I'm not sure about the genetic basis for that. Okay.
But maybe the environment is affected here.
Well, maybe the environment is affected, but I don't know.
For
Let's
Chinese.
move on because we've lost a lot of time with this chit chat.
Yeah.
It's a
Okay.
scientific question, doctor. Scientific question.
Yeah, we'll ask it after the classroom.
After the classroom, we'll leave.
Comes to the telling me so simple.
Did you have a word?
Okay. Okay, so you know Gregor Mendel, famous
Yeah.
guy for genetics, eh?
For the
In the
peas.
1850s, he did these experiments in the peas.
Yeah.
He was a Christian monk. He was doing these experiments, and he
It's a
established.
monk.
Yeah,
It's not science.
no, he wasn't a scientist.
Really?
Yeah. So he established the two laws of genetics, well, that apply to complete dominance. Okay. So we have now understanding that there is a different type of genetic inheritance that we learn from modern, understanding of the DNA. So,
Guys.
so his principles are based on the complete dominance that we just talked about. Okay, so we know recessive and dominant.
Dominant.
This is complete dominance. Okay. We'll talk later on about different type of inherited patterns like incomplete and codominance. So he did in pea plants, and his were controlled experiments. In his experiments, he used what we call pure breeds. So pure breeds, like they have the phenotype, but they have the genotype is homozygous. Okay, so a pure breed will have the same alleles. All right, they will not be heterozygous, and that's how he established these experiments through these breeding pure breeds. Okay. So he tested many things, the shape, and you will see in the experiment, we're not going to do peas, but we're going to work on corn in the lab. You'll see this wrinkled and smooth shape. He did the same thing with peas. He looked at the shape, whether they're wrinkled or they're smooth, the color of the seed, the flower color, height of plant. So he did this, so he got the purebreds and crossed them with each other. And if you do the Punnett square, you'll know the monohybrid cross is very, it's all, you probably studied this in high school, monohybrid? No? Okay, we'll talk about it today anyways. So, based on his experiments, he found out that what these factors are, that he didn't understand them before in that time, because we didn't have those modern genetics. We didn't have the structure of the DNA. We didn't know that DNA makeup controls the activity of the cells. So he called them factors instead of genes, because there was no basis for
Didn't
that.
discover.
Yeah, it wasn't discovered that time. So those hereditary factors like color of the pea,
Size.
the shape, the height, all these traits, he called them factors. Okay, and he said that. So this is his law of segregation. Okay, that the these factors, which we know the genes and alleles, they separate during gametogenesis, okay? Remember the formation of the gametes?
Yes.
We have in humans, remember, in meiosis 1, in metaphase 1,
The chromosomes.
23 chromosomes line up, okay? Pairs,
Yes.
homologous pairs, okay? So then they separate. From 2n we reach n, so we get haploid. So the factors that he's talking about, this is where they separate, okay? And then they come back together, they reunite after fertilization. When you get the sperm and the egg coming together, the factors reunite again. And this is what he established from his experiments. So the first experiment he did was a monohybrid cross. Monohybrid means he's only looking at one trait here, okay? So one trait. For example, it could be the pea color, shape, the plant height, whatever, okay?
Just one.
So in monohybrid cross, you're looking at a single trait, a one trait, okay?
Just the color.
For example,
the
color, okay?
Okay.
So in this case, let's have a look at the color, all right? Now, as I said, he used purebreds, okay? So in the first cross, the parents, they're purebred, okay? Pure green or pure yellow. And so yellow and pure crossed, okay? So when he did the experiment, okay, he found that all the plants are yellow. What does that mean?
Yellow is dominant.
Yellow is dominant. Now you understand what
Dominant.
dominant and recessive is? Okay? Because you crossed a green with yellow, but you never get any green.
Just the yellow.
Just the yellow.
So it's dominant.
So the green color, the yellow color is dominant over yellow.
Yeah.
So all of the F1, we call it, the first generation or familial one, okay?
The genotype for the yellow for the first one is A
We'll
capitalized.
talk about the genotype in the next one, okay?
Okay.
Because here he's just establishing dominant and recessive phenotypes, okay?
Yeah.
It's based on phenotypes. He did so. We just look at in the next slide. I think we talk about phenotype. You have a look.
Oh, the other one. Yeah. Yeah.
Here as well. Okay, we'll look at it. This is the experiment he did here. We'll look at it. Okay. All right. So the F1, all of them were yellow. So that means that the yellow phenotype is dominant over green. What he did was he took some of the offspring, okay, of the first generation and crossed them with each other. And he found out that he gets this ratio all the time, where one of them is green and 3 yellow. So when you do a monohybrid cross, you always get this ratio in the F2, okay? 3/4 is yellow, okay? And 1/4 is green, all right? So when you start off with pure breeds, you cross them the first generation, you get all yellow, and then the second generation, the green appears again, but in a ratio of 1/4, okay? So this is the ratio that you see.
And doctor, the green now is recessive.
Yes. We'll talk about this ratio in a minute, okay? So always in a monohybrid cross, in the F2 you get this, what we call 3-to-1 ratio, okay? So 3/4 is the same as 3 and 1/4, 3-to-1. You understand that?
Yeah.
How many are yellow? 3. How many are green? 1. So 3-to-1.
It's obvious, guys.
It's going to be
All right. So we can use this Punnett square to determine the genotype and the phenotypic ratio, okay? So these ratios we can, so the ratios of the genotype and phenotype can be found using these Punnett squares. So if you have a look at here, remember the gametes, the factors, those alleles segregate during gametogenesis. So we write them. So if the female is capital A, small a, we write in the gametes will carry only one of those alleles because they separate. Remember? So the gamete will contain only one. So we write in this egg, for example, dominant A, and this one, small a. Okay. In the sperm, the same thing will happen. Capital, small. So when we do the process, we go, if this sperm fertilizes that egg, you will get dominant. Okay.
So four chances.
4 different chances. Okay. So this egg, this sperm with that egg, you get that phenotype and so genotype and so on. Okay. So a Punnett square can give us all the possible different genotypes of the offspring.
Doctor, what's the difference between this one? Dominant A, recessive A, dominant A, recessive A.
Where?
This one and this one.
This one and this
The
one?
corners.
Huh?
Yeah.
There's no difference. This is giving you all the possibilities.
But this is.
It's giving you
50 to be like this.
Yeah.
So,
So when we look at that, it gives you the possible alleles in the gamete, okay, and it gives you the possible combination, okay. So the Punnett square is there to show you the gametes in the male and in the female in the other axis, and then you cross. When you cross these, it'll give you all the combinations, all right, that you get from that. So in this case, we got these 4 different combinations.
So doctor, each one, sperm and ovum, have one gene.
Yeah, one allele, okay.
So it's 3 different
One allele.
combinations.
And how many genes?
Well, in reality, yes, but you do it like that because if you say 3, you can't do the ratios. We got to get ratios. Okay. Let me explain it on here to you. You confuse yourself.
Ask you to
Because we're looking at chances of possibilities, okay? So let's have a look at that first cross with the P color, okay? So yellow was dominant over green. So you see he started off Mendel with pure breeds, okay? So both of them are homozygous, okay? So pure breeds, homozygous, okay? With a homozygous recessive, homozygous dominant recessive. He crossed them. When you do this, when you do that, you get 100% heterozygous dominant. Okay. So remember that. In the F1, you don't need to do a Punnett square to get the results here. Okay.
Yeah.
All of them are. Everybody understand why that is the case? Or you want me to do the Punnett square and show you? Okay. So he took this first one and crossed them with each other. So remember, they're all heterozygous dominant. Okay. Come here. Come here and do this one. Hurry up, we haven't got much time. All right. So put the possible gametes. The gametes. Put the possible gametes. The gametes. The gametes first. Let him do it.
do it, guys.
He knows what he's doing. He did very well in the exam. Let him do it.
يمين واي سمول يمين لا يمين واي سمول يمين مربع على اليمين
Hamad, use the gamma.
هنا واي سمول مربع على اليسار مربع اللي اليسار مربع
It put you under stress? You need to work under stress sometimes, don't you?
Yes.
Exam is stress time, huh? You should be working very well under that.
Okay, why capital?
خلاص خلاص كذا كويسة.
You know there's gonna be questions like this in the exam.
It's so easy.
Or small. Yeah, or small, or small. We're gonna do two boxes. Okay.
What's the ratio?
All right. Let's see. So capital Y, small y for one of the parents, the gametes. The other parent, capital Y, small y, because both parents are the same, correct?
Yeah.
Now, if you capital with capital should give you two capitals, huh?
So dominant.
Huh?
Dominant.
So dominant. What? Homozygous or heterozygous?
Homozygous.
Capital Y, small y.
Dominant heterozygous.
Dominant heterozygous.
Also
Small y, capital Y.
dominant heterozygous.
Yeah. Small y, small y.
Recessive homozygous.
Come here. Give me the genotypic ratio. The genotype. How many different genotypes do you see here? How many genotypes do you see here? Write the genotypes. Write the genotypes. The genotypes.
Genotype. 3 genotypes.
How many genotypes? Here, use this. Don't color your
3
hand.
genotypes.
Quick.
Genotype.
Genotype. We're already over time.
Yeah, it's okay. It's okay.
Number.
أول شيء قال لي أول شيء أول شيء قال لي
This is 2. This is 1. This is the genotypes.
Yes. Now, what is the? So this was the genotypic ratio, okay?
Yes. Because there are 2 dominants.
So there's 3 different genotypes here, huh? There's 3 different genotypes. Now, what's the genotypic ratio? How many different genotypes are there? Phenotypes are there?
The phenotypes 2.
Wipe this one. You got it in the wrong place, so people don't make.
So what's
I want the. This is the genotypic ratio. You just wrote it here.
okay?
I want the phenotypic ratio. Not there. You're writing in the genotypic ratio.
Okay. So what about here?
I scared you. This is how you should be working under pressure like that.
Yellow.
Come on. All right, you got it wrong. Sit down. Okay, I'll tell you what it is. You had it right before then. How many different phenotypes do we have here?
3. No.
2 different phenotypes.
I mean genotype.
This and this are
The
what?
same.
Yeah.
Dominant.
So what are they?
Yeah, yellow.
Yellow. Okay, that's the phenotype. 3 yellow. How many green?
1.
1. So the ratio is 3 to 1, yellow to green. You got that? Simple as that. And that's time we can. I'm 2 minutes over your time.
It's okay.
Y capital is dominant, okay?
No, it's not heterozygous.
It's heterozygous.
Homozygous at the same time, or? Yeah, just heterozygous.
What do you mean?
For Y capital and Y small, it can be homozygous and heterozygous at the same time. No, just heterozygous.
Heterozygous, come on. Heterozygous.
Homozygous.